Okay, I think most people are here now, so we'll get started. So thank you everyone for joining today's webinar, exploring the exciting new partnership between Atelerix and Cherry Biotech, and looking into how this collaboration is transforming the way advanced 3D organoid models are transported in research. My name is Sarah Marsh. I'm from Atelerix. I'll be the host for today's session. And today's session offers a scientific insight into both Atelerix's preservation technology and Cherry Biotech's organoid platforms, and provides the opportunity to hear directly from all the experts that are driving this collaboration forward. So I'm delighted to introduce our speakers, Dr. Mathilde Cadoux, research scientist and immunology project leader at Cherry Biotech. Pierre Gaudriault, Chief Business Development Officer and co-founder at Cherry Biotech, and Dr. Steve Swicklow, Chief Scientific Officer and co-founder of Atelerix. So following the presentation, we'll move into a live Q&A session where Mathilde, Pierre and Steve will help to answer any questions you have, and we can go through those. And before we begin, just to note that any questions that you do have, please submit in the chat during the presentation, and we will collect them and address them in the Q&A. So without further ado, I'll hand over to Mathilde and Pierre to go through the presentation.
All right. Thank you very much, Sarah. I cannot see the slide at the moment. Yeah, I know it's not in my hand. All right, perfect.
Thank you.
Okay. So thanks. Good morning and afternoon, everyone. So today we also would like to thank Atelerix for the opportunity of presenting the results of the ongoing collaboration, and particularly on how the combination of our technology has actually enabled the worldwide delivery of ready-to-use complex structure like organoid. So on the next slide, I wanted to start with a little bit of context in order to walk you through why we have developed this and what's the need behind this development. I'm pretty sure most of you know that during the drug development, there is a high failure rate when drugs is actually validated in preclinical phases at the moment it starts to be tested in humans, so during the clinical phases, which is particularly true in phase 2 by a lack of efficiency. On the other hand, it means that the classic preclinical model, either animal or let's say 2D cell culture, are not predictive enough of what would be more likely observed in the human later on during the drug development. And similarly, in the hospital, in particularly oncology, there is a lack of predictability in which cancer that is treated will actually induce a relapse. And we have actually an overall stat of more than 1/3 of the cancer will actually relapse after 5 years. So it seems again that it might be beneficial in the clinical use to have tools that allow to actually predict what will happen over time when a patient is treated with certain drugs. On the next slide, in the few last decades, there is a promising technology that has emerged that was organoid chip or organoid, which is actually 3D cell culture. It starts from labs, as often with really nice publications, and then it starts to transfer into industrial world, encouraged with a push of the regulation, particularly in the US, with the two consecutive FDA Modernization Act 2.0 and 3.0. The Europe is actually following up on this path, and sadly, we are still observing on a pure demand and market point of view, a low adoption rate of this technology. So we ask ourselves why, and it seems that they still have scientific and technological bottlenecks to be lifted. In the next slide, One of them is what do we consider something that is usable in either hospital or in the pharma. In our own cases, we want to develop a solution that is a platform, a hardware part, associated with a protocol, let's say the recipe, and a biological model for a specific context of use, for a specific usage of the system. In the next slide, if we take the whole chain of events, the problem is that the industry, the researcher, and the final user will have to switch from what they have used in decades, particularly 2D culture, toward 3D culture. And actually, this transition takes time. It might be perceived risky because you don't know the protocol, you don't have the know-how or the knowledge. So you need to have access to ready-to-use solutions. If we talk about organoids, it means having access to ready-to-use organoid that you can actually handle directly in your facilities. In the next slide, and this is exactly what we are here to talk about today. It's how the solution of Cherry Biotech, which is called the OrganoidPlates, is an assay ready to use standard multi-well plates, can be shipped worldwide in combination with Atelerix technology. And I'm giving the floor to Steve to continue and present to you their contribution.
Thank you very much, Pierre. As Pierre mentioned, these complex models can be instrumental in really pushing drug discovery and allowing the replacement or at least part replacement of a lot of the animal models that lead to a lot of drug attrition. But the problem really with these models is generally if you increase complexity and physiological relevancy, you increase fragility. And this then makes it very difficult to ship those complex models, including organoids. The survivability and shipping stability decreases. Results in high shipment failure and model variability, and also reduces the accessibility and really harmonization of advanced models that is really necessary to push their full implementation into drug discovery and preclinical development. Next slide, please, Sarah. And Atelerix is really positioned to say that it doesn't have to be this way. On to the next slide. We provide cryo-free, hassle-free ambient logistics solutions that enables fresh sample shelf life to be amplified from hours to weeks. What this means is that we can provide technologies that actually enable the shipment and distribution of these complex models so they can arrive at the testing sites where they are really needed. We supply biosample preservation kits, which are quick and easy to use. And what these kits do is maintain sample viability and functional performance, and importantly, integrity of the models or biospecimens that are being shipped, enabling complex tissues, organoids, blood, sensitive cells, and even therapies to be shipped. Our products are easy to validate. They de-escalate shipping. They are a lot easier than, say, looking to ship consignments using cryopreservation. And they can even slash costs by up to 10-fold compared to cryopreservation. So really enables a flexible, low-cost supply chain and workflow from sample collection to final application. Next slide, please, Sarah. And to do this, our platform technology relies on specific formulations of alginates that offer ultimate cytoprotection during hypothermic storage. Hypothermic storage is anything below 32 degrees. Commonly, if you're looking at hypothermic storage, it's either room temperature or refrigerated. And usually at these temperatures, cells tend not to survive for too long, predominantly because of damage to the lipid membranes and other structures of cells. What our formulations of gels do is stabilize cell membrane integrity, allowing samples to be held at room or refrigerated temperatures for extended periods. Because we're working on the lipid membrane, we're protecting that lipid membrane. It means that we can apply it to everything from fresh cells, cell models, tissues, and viruses, allowing them to be stored for longer with no deterioration in quality. Next slide, please, Sarah. We also formulate our gels to have optimal mechanical protection during shipment. This is particularly important when you're looking at shipping organoids and complex models, because you need to protect shearing from liquid movement and also buffer those mechanical forces, those vibrational drop and shock forces that can cause damage to those biological samples. Our gels enable that for the safe transportation of the biological material, either in vials or in multi-well plates, which is the topic of this webinar. Next slide, please, Sarah. And really just to kind of set the theme for the next section is really, we were looking with Cherry to utilize our product well ready for in-plate preservation, to stabilize ready-to-use organoid plates during storage and shipment to ensure model integrity and function is retained to enable distribution. And Mathilde is going to continue on from me to really describe the case studies that Cherry Biotech have gone through to utilize our technology for that global shipment. And with that, I'll pass on to Mathilde.
Thank you, Steve. So, like Pierre said, we've developed some ready-to-use organoids in plates. I will talk more about adipose organoid and cancer organoids. But thing is that it's great to have ready-to-use organoids, but how to ship them to clients? Next slide, please. We all know that worldwide delivery is a logistic nightmare. Next slide, please. Because there's two main possibilities to send biological samples. It's either freezing, it's not that simple, and the cold chain logistic is complex and expensive. And of course, there's no go for certain models like our adipose tissue model organoids, because it decreases viability when you freeze and thaw them, and it can go to burst, meaning the cells can explode and the oil is released inside the medium. Next slide, please. The other, when we were looking at the shipment, the other possibility is our portable incubators. Thing is that it can only handle a few plates, I think maximum 5 plates. Also, most organoids need refreshments of medium each 2 to 3 days because we have a lot of cells inside each well. And so even a small delay can compromise the cells and their viability. Of course, this kind of portable incubators are really expensive, and it's because it's expensive that we want the incubator, the box back. And so the logistics of return of the incubator add another level of complexity. Next slide, please. So when we found Atelerix, we were quite excited because it could allow us to ship ourselves at room temperature, cutting costs, of course. We are a small startup, so we don't have any money. We have less logistics, and most of all, we are protecting our organoid viability. So we've done some in-house tests and shipment to clients, and all were really successful. So we are really happy. Next slide, please. So first of all, I will talk about the adipose organoids, the ready-to-use that we've created. Next slide, please. So we've created adipose tissue model starting from AGSCs, which are great and really described by Lonza, or SVF in standard 24 multi-well plate. Next slide, please. We have some, of course, quality controls. First of all, microscopy. It's allowing us to visualize the lipid content and allowing us to see through the development of the adipocytes. We also have adiponectin production, which can basically allow us to know how much differentiated the adipocytes are. Same for lipolysis assay. And the last thing is the in-house automatic measurement tool, which is a pipeline allowing us to measure organoid diameters during differentiation, so all along the experiment and right before shipment, to assess the evolution and the homogeneity of the organoid production. It looks like this. So we send that to the clients. We can see that there's all the wells from the multi-well plate. We can see that the different variability of the size of the organoids. Next slide, please. So this pipeline with the ADST-based organoids, we follow them with a protocol, including induction medium and maintenance medium. We could follow the spheroid diameter, then we let them grow and differentiate until day 24, then embedding them into Atelerix hydrogel for 4 days, and then let them out, dissolve the hydrogel, and then let them in culture. And as you can see, there's no really big difference in the size, which is quite reassuring because it means often that the organoids is not dying. Next slide, please. And it's true because as you can see or not see, there's no difference between before and after Atelerix dissolution, which is quite reassuring and convincing. Even after 4 days in the hydrogel. Next time, next time. Then we've done 5 days after the Atelerix dissolution, we've done some stainings, so the lipid droplets are in green, and you can see that it's full, the organoids are full of lipid droplets. There's no necrotic core, so the organoids are really in great shape. Next slide. Then we've done some also in-house tests this time with SVF. Next slide, please. And again, with the pipeline, we could assess the spheroid diameter, pre-Atelerix, so the green, then day 0 during just after the dissolution of the embedding, the hydrogel, and 5 days after. And you can see that 5 days after, the diameter is increasing. And you will see that this increase is a good thing. Next slide, please. Because after 4 days, you can see that the lipid content is increasing 14 days after embedding. So it has something to do with, of course, the differentiation, but we think that probably there is a beneficial effect of the embedding. So this is 4 days, and the next slide is 7 days. We've tried that because we had some, let's say, a little bit of problems because of a storm that delayed some of our deliveries. So we wanted to know if even after 4 days in Atelerix, it could be still okay for the organoids. And as you can see, it's still okay, and the lipid droplets are really big, and the organoids don't show any necrotic core. Next slide, please. So, adipose organoids with Atelerix, even through a storm, they are really great. From 3 days to 7 days in Atelerix, they're in great shape. They're potentially, and this we need to verify, there's a beneficial effect of the embedding, especially with the SVF-based organoids. And all the shipments that we did to clients are really successful. Next, please. So the next use case are the cancer spheroids and organoids. So we have a large catalog of models via our partner Cytian. We can produce scaffold-free spheroid or organoid cancer models. So really simple or really complex. So with increased microenvironment and with ECM, so extracellular matrix. On top of it, including fibroblasts, endothelial cells, macrophages, and other type of cells. Next slide. So The example is the triple negative breast cancer spheroid. So we can see that again with the pipeline with the spheroid diameter before shipping. So right after doing the Atelerix, before the Atelerix embedding, you can see that there's not between the before and after. I mean, with the 5 days of shipping, you don't have many differences between the before and after. So it means that the cells are not growing, so they are stopped in their metabolism. So it's great. Then after the relieving protocol, the dissolution of Atelerix, you can see that there's an increase of the diameter, meaning that there's proliferation, and the organoid didn't die. Next slide, please. So great proliferation and viability after dissolution of Atelerix for the breast cancer model. Next slide, please. Also, as you can see here, we've done some tests. So 4 days in Atelerix. The control incubator on the left is showing cancer organoids without any embedding with calcine and PI. So like that assay, you can see that there's no or low amount of dead cells. And it is the same with Atelerix on the right, or really low amounts. Next slide, please. What we've done is to push the complexity even more with way more complex organoid model. So with organoids of cancer with TME, so an embedding of all the cells that I mentioned before, fibroblasts, CAFs, macrophages on top of the organoids. So it's over 1 millimeter of collagen hydrogel on top of the cancer cells, including many TME cells. And you can see also that there's no cell death. So to conclude, I think we are really happy. Atelerix has become a strategic partner for Cherry Biotech. Our collaboration unlocked the possibility to deliver worldwide, which is huge, ready-to-use organoids, and also the simple protocol and short recovery time allow scientists and so our clients to save time and generate more relevant and consistent data. That's it for me. Thank you very much. I'm happy to answer questions.
Thank you so much, Mathilde and Steve, for that wonderful presentation. We will now move on to the live Q&A session. Thank you everyone who has submitted questions so far. Do put any additional questions in the chat now, and we will go through them first. So our first question, can assays be done in shipping medium, or does a full medium change need to be done after being shipped?
Well, I can probably start the answer and Mathilde can complement or Steve based on the protocol. So the way we are doing it, we don't have at the shipping necessarily an extracellular matrix. So we basically embed the organoid directly in the Atelerix hydrogel. And when the customer is receiving it, there is a relieving and a dissolution protocols that you basically follow the step. It's really easy, really quick. And then you place the organoid inside the maintenance medium or the culture medium that you will use afterwards.
Yeah,
thank you.
Thank you,
Yeah.
Pierre. Yeah, just to add to that, there are certain downstream assays where you can have the gel still in place above your in vitro model, be it a model A or 3D model. But it is restricted to certain downstream assays due to interference of the gel being in place with those downstream assays. So for most applications, it's quite a simple process of adding a dissolution buffer and then removing that dissolved gel and replacing with medium again.
Thank you, Steve. We have another question. Do you know if the shipping medium is compatible with neurons?
I'll take
you.
this one, yes. Yes, it is. So we've had a number of different people that have utilized well ready for the shipment of neurons, cortical neurons, DRG neurons, quite a few different types of neurons. And really, the well ready system, it's one of those kind of stress tests for that system because neural networks are incredibly delicate. If you're trying to ship them in just medium, they just shear straight away. We've had great success in protecting those neuronal networks and maintaining the viability and function of neurons.
Thank you, Steve. Now, next question. Are there other cell lines that you've tried to deliver with the gel?
In our case, we have a whole range of things that have already been shipped. So Mathilde mentioned ADSC, SVF for the adipose tissue. We have the 4 subtypes of breast cancer, lung cancer. We have some liver model, and we are actually currently developing colorectal cancer as well. That's kind of the off-the-shelf cell lines we have. Then we have also a capacity of making them custom based on the cell lines from our partner Cytion and all the primary cell that we have access from Lonza as well.
Yeah, and
Thank you. Yeah.
for the general utilization of our technology, I think it's up to about 150 different cells, biospecimens, tissues, viruses that have been successfully preserved using our products. Now, there's a comprehensive list on our website if you wanted to have a look.
Thank you, Steve. We have another question through regarding cost. Can you tell us about cost reduction in terms of percentage for shipping cells when compared to something like a mini liquid nitrogen tank?
Mini liquid nitrogen tank. That's even more expensive than our general cost comparisons. Usually we compare with dry ice, and what you can achieve utilizing our technology is a reduction in payload weight. The size of your package from a big 20-kilogram box of dry ice down to what is essentially a 1 to 2 kilogram box. That reduces shipping substantially. There's no requirement to source those refrigerants, and the actual shipment costs, because you're not shipping with the hazardous refrigerants, is also lower. So we estimate a 10-fold reduction in cost normally. If you do have a biological sample that actually requires a dry shipper or mini liquid nitrogen container, I think it was said, those costs can be incredibly inflated. They can be hugely higher, and there are a lot of airline restrictions on actually using those dry shippers. A lot of routes won't take them.
Perfect. Thank you, Steve. Another question for Cherry. Did you measure any delay in development between shipped and non-shipped organoids?
No.
It's the short answer.
No, and the only thing I can at least answer for the adipose tissue, we need to have like 20-25 days of differentiation, and the 4 days inside Atelerix didn't affect anything.
That's great. Thank you, Mathilde. Next question. Have you tested patient-derived cell suspensions? So not yet established organoids, but the primary cells.
Yes, we did. Not completely shipped because there is a lot of, ethical restriction, etc. But in our internal R&Ds, yes, we did. the base of the plate relies on micropatterning that will promote the self-assembly of and differentiation of the cell's contents. So for patient-derived, we basically start with after a dissociation of the fresh tumor or the PDX or whatever the patient-derived materials we have, and then with the protocol we have developed inside this specific micropattern plate, we have a spontaneous assembly of or recapitulation of the original tumor. Then, as Mathilde mentioned, there is, depending on the tumor, on the aggressiveness of the tumor, on the proportion of different cell types, there is variability in the time of differentiation to have something that is actually quite similar to the patient sample. The histopathological data that we have are fairly similar between the organoid obtained after, let's say, 15 days of differentiation and the original tumor.
Thank you, Pierre. That's great.
Just to add to that, Atelerix also provides solutions for starter materials for patient-derived organoids and complex systems, so both dissociated cells and the tumors themselves. So provide tissue-ready and also cytostore. Cytostores for cells, tissue-readies for tissues.
So that's one. Go on, Steve.
Yeah.
No, it's
Thank you both.
In patient-derived material, the transportation, if it's fresh, is fairly challenging. We are based in Paris. If we have a sample coming from Marseille, the time of transportation can really be deleterious for the viability of the cells, the heterogeneity, etc. So having solutions that kind of guarantee that everything is quiescent and will arrive in good state is also a guarantee that the further experiment will be made in a good standard protocol.
Perfect. Thank you both. We have a couple of questions regarding the lid. So what type of plate lid did you use? Was there a special lid for the plate that does not allow leaking?
During the transportation, you mean? I guess.
Yes.
Well, I guess Steve, you can go, yeah.
Yeah, sure. Within the kits that we provide for in-plate preservation, we provide a sterile plate seal that will just go over the top of your wells in your plate, and then you can just place a standard culture lid on top. Because it is a gel, you don't get sloshing of medium like you often would if you're trying to ship something in medium, but why would you? The lid is just standard, so you don't need to add anything extra.
Perfect. Thanks, Steve. We have another question. Can we explain a little bit more how the gel works to preserve the TMEs during shipment?
I guess that's for you, Steve.
I guess it is. That's more mechanism. So really, so the basis of hypothermic preservation is if you're trying to store either a cell or a multicellular organoid, you go through a phase transition in cell membrane kinetics when you take it from the incubator to room temperature. The room temperature actually enables the not quite metabolic arrest, but it enables the cells to have their metabolism reduced to such a level that they're in a state of suspended animation. This theoretically allows them to be stored within sealed receptacles. But because you are changing the lipid membrane from a liquid state down to a static state, which is just a phase change going from 37 to about 20, this results in pore formation, which results in uncontrolled ion entry both into cells and also the extracellular matrix within organoids. This causes continual destruction of the lipid membrane and swelling of the ECM, which results in death of that organoid. Our gels are designed to essentially act as a solid state sponge to prevent that dysregulated movement of water and ions and other small components into the cell, thus preventing their swelling and destruction. That's the core mechanism by which our products behave, and that is how they have protected Cherry's excellent complex organoid platform.
Thank you, Steve. So I think we've got time for one more. Did you try to test the accessibility of different drugs with organoids, with or without the Atelerix's kit?
Yes, but they usually do not happen at the same times, meaning that the Atelerix hydrogel is really for the transportations. So all the drug test would have happened either before or after. So we never actually tried a kind of drugs penetration, either biologics or small chemical in the hydrogels, because in the way we designed our product, it's not meant to be used at the same time.
Wonderful. Thank you, Pierre. So thank you everyone for submitting those questions. It's great to build on the discussion. Very special thanks to Mathilde, Pierre and Steve for sharing all of your expertise and providing this deep insight into the collaboration and how we can improve accessibility, reproducibility and scalability in organoid workflows. If anyone would like any further information about either of the technologies or would just like to continue the conversation a bit further, please don't hesitate to reach out to any of us. And thank you again for joining, and we hope to see you on our next webinar. Enjoy the rest of your day, and bye-bye.
Thank you very much all. Bye-bye.
Bye.
Thank you.
Goodbye.